Welcome to Dan Sains from NYU. He joins us as new MCWG member representing NIDCD Council. And Dan's been a leader in the field of central auditory development and plasticity. So I've already had a chance to speak with Dan. I think Dan, you will give us you will give great contributions to us, and I really look forward to working with you. On a somewhat less happy note, we're saying goodbye to David Tank today. David has been inspirational to the field and to brain in particular. I think his science needs no introduction here. David has been involved with the Brain Initiative since its birth. He was a member of the original ACD work group that gave rise to the Brain 2025 report, a visionary strategic plan for the initiative. He saw Brain Initiative through its infancy, childhood, adolescence, and I think young adulthood right now. David, as with any good parent, you know that your job is never done, but we are letting you off for time served after 6 years of great insight, input, and wisdom. But I hope I can call on you for advice as we need it going forward during this exciting next phase, which I would say, quite honestly, you really helped get us to this place right here. So thank you very much. I really can't overstate my appreciation for all that you've done for us. And again, I hope you will take my phone calls after today. I've had the privilege of working with a great staff. Andrew Beckel-Mitchener is our acting deputy director in the Office of the Brain Director. Greg Farber and Ned Talley have been serving as senior advisors, and we have a great staff as well with Michelle Pearson, Nee Floyd, and Shauna Bach, and a very talented AAAS fellow, Ryan Richardson, who you may be hearing from time to time. And then it really does take a village. Over 100 people at NIH pour their hearts and soul into this effort on a volunteer basis. They do it because they want to do it and they love it. We have 8 project teams, coordinating team members, a vast number of other support staff that really make things happen for us and for you. Now we have 8 project teams, and you'll be hearing from Team Thai later today. Team Thai actually was christened as Team TD for Training and Diversity, but found a much better and better description of what they do. And now they're known as Team Thai, or Training, Inclusion, and Equity. And the co-leads for that are Jay Churchill and Michelle Jones-London. So I hope you'll join me in looking forward to their presentation after I get through my introductory remarks. Now, COVID is, of course, on everyone's mind. How can it not be? I just want to remind all of us that as the situation continues to evolve and hopefully gets us to a better place, you can see on the NIH's website what we are doing to help out researchers that are getting into a bind because of the pandemic. And Brain is handling issues on a case-by-case basis, but please visit this site, talk to us, come to the Brain website. If you're a Brain awardee, definitely talk to your program officer if you're having issues with the progression of your project. We've also granted a 2-receipt cycle extension for now for eligibility for prospective applicants. So again, we're doing what we can to help out the extramural research community, and we're especially concerned about our trainees and more junior investigators. And we hope that for now, this extension will help alleviate some of the stress around eligibility issues. Now, there is opportunity for the neuroscience community to address the COVID crisis, and I just wanted to point out to you some funding opportunities specific for COVID-19 that might be of interest and relevance to folks in the neuroscience community. So just, you can take a look on the NIH website. I don't have time to go through these in detail, but just want to draw your attention to the idea that we can contribute to this terrible pandemic and hopefully help us see it through and get out the other end in a much better way. Okay, so how about some good news? The good news is we received for Brain an increase in our appropriation for this year. It winds up being a net increase over fiscal year 2020 of $60 million. And that came about due to a $100 million addition to our base funding. So that, together with $100 million in CURES Act funding, brings us to a total of $560 million to invest in fiscal year 2021. And recall that the Brain 2025 report recommended that $500 million would be a good place to be. and it has been a good place to be. And this kind of sets the stage for moving ahead. You'll notice that if all goes to plan, and that's an assumption, in fiscal year 2023, we'll have a big increase in funding because of the Cures Act funds, which right now is projected to be north of $900 million. And it was with this increase in mind that we had last year prepared ourselves for launching transformative projects. As you may recall, we decided to move ahead with 3 large projects. And I just wanted to update you on where things stand, especially in the context of this funding outlook. So the additional funds will enable the accelerated launch of, in particular, this armamentarium for cell type specific access tools. And here again, the goal is to develop tools for cell access in rodent and non-human primate brains, human cells and tissues, with the long-term blue sky goal of developing new therapeutics for human brain disorders. The first RFA is issued. Receipt date is coming in a couple of weeks in February. I hope those of you that are writing your grants are getting there. There's another receipt date in October. We're very excited about this. $20 million of the additional funds we received this year are earmarked for the armamentarium project. We've talked a bit about the brain cell census project. Based on the progress in the field, we are planning to launch a larger project focusing on the human brain to develop a comprehensive human brain cell atlas. And again, $40 million of our increased funding this year are earmarked to help us accelerate the launch of that project. And ongoing are discussions about developing next generation technologies for brain microconnectivity analysis, the wiring diagram or connectomics project. And we are moving ahead on the planning phases of that. So what else are we up to this spring? We have a lot of workshops coming. We have a series of 5 workshops focusing on brain connectivity that will address the state of the art in mapping complete circuits and opportunities for advancing the field, challenges, and also what will it take to map an entire mammalian brain or mammalian brains at scale. We have been in discussions with the Department of Energy's Office of Science, and we're pleased to say that we're co-hosting this workshop series together with the Office of Science. So you can go to the BRAIN website and get more information. I do encourage you to dial in and attend. This is, of course, all the conferences I'm about to describe to you will be held virtually. Next, we have a human brain cell census workshop that's being put on by both NIH Blueprint and BRAIN. And this workshop will focus on tissue quality and collection and processing pipelines for human brain samples. This is the basis. This will form the basis of standing up a successful comprehensive brain cell atlas project. And I would like to just underscore that many of the issues that we discussed about ensuring diversity in our human sample cohort were taken into account in the planning of this workshop, and I think we will see some measurable impact from the discussions we had last time. And then finally here we have 2 workshops on non-invasive imaging technologies. The first dealing with the topic of disseminating technologies that were developed under BRAIN Initiative Awards, and the second will be on identifying potentially revolutionary technologies. That workshop will be held in March. And of course, the big event, we forgot to put a slide in, is the BRAIN Investigators Meeting, which will be held June 15th through 17th, again virtually. You'll be getting lots more emails about that. Okay. The BRAIN data sharing policy is now in effect. So all the applications that are about to be funded will have to comport to this new policy. There will be a requirement to submit data to one of the BRAIN data archives. There needs to be a specific resource sharing plan that's approved by BRAIN program staff, and we are allowing costs to facilitate this data sharing. So you'll be hearing more about this as we roll along. Program staff will be working very closely with investigators to make sure that they can meet these requirements. So I just wanted to take a couple of minutes, just a few short minutes, to highlight 2 scientific advances that I think are pretty cool and really point to directions we can go to in the study of neural circuits and their role in behavior. Here's a study that was recently published by Lin Tian's lab at UC Davis. And Dr. Tian has been a pioneer in developing sensors for neurotransmitters. And what's really quite remarkable and notable here is that based on the previous work that she's performed and published on developing sensors for biogenic amines, here we have the so-called RD light or red light, which is a sensor for dopamine, but it's shifted into the red end of the spectrum, which means that it can be used simultaneously with other reporters such as GCAMP to sense simultaneously calcium levels as well as neurotransmitter dopamine. And it's usable using fiber photometry. So this really opens the way to study the dynamics of neural circuits involved, for example, in movement disorders, reward and addiction, by looking at both neurotransmitter levels as well as together with the output that's occurring in the circuit. So I think this really opens up a lot of really interesting new avenues for research. Very recently, Nanthi Suthano's lab has published this paper in Nature. And what I really like about the study is that it allows the analysis of neural circuits behavior in what I refer to as awake behaving humans. So here, this is a great example of our research opportunities in humans where there are a cohort of patients who are implanted with a NeuroPace RNS device, a bidirectional electrode to treat epilepsy. And we're using this now, or the Suthano's group is using this now to record activity from the human subjects who really are research partners very clearly, and to send these data to a backpack, but also to simultaneously monitor other biomarkers such as their motion, their heart rate, skin conductance, to measure perspiration, respiration, eye tracking, together with scalp EEG, and also global positioning. So they can actually monitor the neural activity that occurs while a person is walking and navigating a space. And what they found was a high theta band power in medial temporal lobe when subjects navigated to a boundary of the room. And what was really quite interesting was that a similar activity pattern was found when the subjects observed another person navigating to the same spot in the room. So this kind of gets toward the notion of representations of both self and other. And I think while limited because of the number of patients that can be studied, this really does open up a new vista for studying the role of neural circuits and, again, awake behaving humans. Okay, so today's open session will be focused on the role of brain and NIH in advancing diversity, equity, and inclusion in the scientific workforce. And I'm just going to go through a few slides to set up the discussion. We'll be hearing from Team TIE about the brain portfolio in support of this mission. And in a bit, we'll be hearing from Dr. Marie Bernard from the Office of Scientific Workforce Diversity to hear about the larger efforts that are going on within NIH. And we'll follow that with an hour-long discussion based on these presentations. But let me just walk through with you very quickly some of the things we're doing in brain, other activities in NIH, and I'll try to put that in the context of what's going on in the scientific community. So starting with brain, we released a request for information a while ago, last year, requesting input on scientific and workforce diversity. We received some very helpful comments coming back. I can summarize this by saying that there were calls for us to enhance the workforce. This is a enhanced workforce diversity and inclusion through a variety of means, through novel funding mechanism, increased training programs, broader community engagement. And also highlighted was the need for equity in the grants review process. This has been touched on, I think as Susan mentioned, in several papers that have been published recently on the topic. And we need a transparent tracking of diversity metrics for brain applications and awards. Now, I'll just say it at the outset that I personally, and on behalf of BRAIN, we take as a fundamental premise that diverse perspectives lead to better scientific outcomes. And this is a premise, I think, that's based in pretty good data, good evidence. And I'll just state right now that we are working on ways to actually put these words into action. And I hope to be able to tell you more about this at the May meeting, if not before. On the broader NIH level, there's this challenge program to award prizes to institutions that develop proposals for enhancing and sustaining gender diversity within their institutions. You can find the details here at this challenge.gov website. And again, we'll be hearing from Dr. Marie Bernard in about an hour or so on the efforts that NIH is undertaking. And I think one highlight, I'm sure that Dr. Bernard will take us through, is this new FIRST Award initiative for faculty cluster hires. Now, again, what we're doing is not done in a vacuum, and I'm very pleased to see the scientific community organizing to address the issues of racial and gender disparities in the scientific workforce. And here is the declaration from the ALBA network, affirming that the pressing need for fostering fair and diverse scientific communities. You can learn more about this network and declaration at the website listed here at the bottom of the page. But again, I think the key to effecting change is to organize the community. And I'm really pleased to see that the community is organizing in this way. And this is now extended to the publishing world. Cell Press has issued a very strong statement that identifying and promoting diversity and equity in the teams performing the research, and the research that is being performed is critical to the scientific mission. And they are now instituting a process where the characteristics of the team and of the research vis-a-vis diversity, equity, and inclusion will now be included. And these data will be helped not only to monitor progress, but really to keep all of us honest in our quest, I hope shared quest, toward enhancing diversity, equity, and inclusion in all that we do. So with that, I will stop. I know we're probably at this point, we might be running late, of course, but if there are any questions, I'm happy to take them now, and then we can revisit these as we get to the discussion period later this morning or afternoon.
I don't see any hands raised. Eve, did you want to say something?
Hi, Eve.
Yeah, hi. Hi, everybody. Nice to see everybody. There was a recent paper published that argued that papers with female senior authors did less well in citations. And I was wondering whether anybody was interested in following that up with another study or et cetera. I mean, the effects were relatively small, but clearly noticeable. And 40 years in, it seems like that really should have already washed out as an effect. Adrian Fairhall and I wrote a little news and views type commentary about that. And I don't see that any of the things that Cell Press is talking about are going to deal with that unless they really change their decision-making process.
Right. So I think one argument that Debbie Sweet made in her perspective piece is that we need to start tracking the data in order to be able to find out where we are, and therefore to identify ways we can move the needle as it need be, and also be able to assess whether we're getting closer to where we want to be or not. So Eve, if you could, if you could pop a link to your news and views in the chat, that would be great.
Okay, wait. Walter was asking something about it being redacted. I don't know what that means. Yeah, I will.
Thank you.
John, I had a question about diversifying samples. Really about the scope. One thing that we are experiencing now at the Stanley Center of the Broad, as we try to diversify IPS cell collections, any tissues globally, Sub-Saharan Africa, India, indigenous populations in South America, our national laws and legal restrictions sometimes on the movement of tissues, sometimes only on identifiable personal level data, sometimes on both. And I'm just wondering, about the diversification, you'll put all this effort into brains. How diverse will the collections be? Will you reach out globally? And secondly, is there a way that NIH can help with coming up with agreements to permit tissues and information in certain circumstances to cross national borders?
Yeah, those are great questions, Steve. It's a work in progress. I'm hoping that some of these issues can be, first of all, identified and written down so they don't get forgotten at the workshop. It's actually this is going to be the first of, I think, at least 2, if not more, workshops. We're also engaging other partners in this endeavor. For example, Chan Zuckerberg is quite interested in this space as part of, but not synonymous with, the Human Cell Atlas project. And we'd certainly be interested in talking to you about your experiences as well, and we will be engaging on that. I mean,
Right.
It's one thing to say we want diverse samples, but of course, if true diversity is going to require reaching into sample pools, not just within our borders, and then we're starting to deal with other issues as well. But we will be certainly considering these as issues to navigate.
Yeah, they're really challenging. The human issues are sometimes harder than the science issues.
Yeah, for sure.